Hello guys, how are you? This is Dr. Mobeen again, and we are talking about immunology. Today's lecture is a little bit different lecture. So what we are going to do in today's lecture is that the topics which we have already covered in immunology, we will cover some possible potential USMLE questions. So the very first question, and I'm hoping to cover these topics very quickly so that we can have some high yield concepts coming out of it. So the very first question which I have for you, and if you have studied these immunology lectures which we have studied before, then you should be able to answer that question very quickly. The question is that if we find a few cells, if we find cells which have reduced expression of MHC class 1 molecule on the surface, for example malignant cells, breast cancer, liver cancer, or some other cancer cell, malignant cells. So if some malignant cells are found, they have reduced expression of MHC class 1 on their surface, which immune cell, here is a question, which immune cell will take care of these cells? So again the question is, if we have found some cells with the reduced expression of MHC class 1 molecules on their surface, which immune effector cells will take care of these cells? And if I gave you the choices of the effector cell, if I said, let's say neutrophil is one choice, macrophage is another choice, cytotoxic T lymphocyte is another choice, and natural killer cells is another choice. So again the question is very simple. After some lab analysis of some cells, it is found that some malignant cells, it is found that the expression of MHC class 1 molecule on the surface of the cells is reduced. Which cell is going to take care of these cells? Neutrophil, macrophage, cytotoxic T lymphocyte, or natural killer cell? So let's quickly answer this question. If you have studied the innate immune system, which is very initial chapters of immunology lectures which I have delivered, you should be able to answer this question very, very quickly. If not, let me just quickly give us some context. So the first thing to note, do not forget this, all nucleated cells exhibit MHC class 1 molecule on their surface. So MHC class 1 molecule, if you see, it is normally made up of structure like this: alpha 1, alpha 2, alpha 3 microglobulins, and then beta microglobulin. So that is the structure which is attached to the cell. Actually this is how the structure is, beta. So the alpha 3 portion is the one which is attached to the cell membrane. So anyways, this is an MHC class 1 molecule attached to the cell membrane. So the question is, number one, the first thing, all nucleated cells exhibit MHC class 1 molecules on their surface. That is true for all nucleated cells, all healthy nucleated cells, number one. Second thing, MHC class 1 molecules normally exhibit a peptide fragment in them. Normally this peptide fragment which they are carrying is that of a protein which is part of the body. How does that happen? So if you see how this MHC class 1 is formed, so if you go inside the cytosol, if you go inside the cell, you would find out that proteosomes, there are proteins which enter the proteasome. So let's say this is a proteasome. Proteins enter the proteasome. So this is really, let's say if we blew up the cytosol and we're looking into it, and here is a proteasome, there are protein in there. Proteasome will break up the proteins and bring the fragments out. So this is a protein, the protein is broken up and the fragments are taken out. These go into the endoplasmic reticulum. In the endoplasmic reticulum, these fragments are loaded into, so how do they go in? This is a very important question as well. TAP1 and TAP2 proteins, the transporters. So this in itself is a question that, hey, if somebody has a genetic problem and the TAP transporters are not working, what's going to happen? So we'll cover that question at a different time. But anyways, these fragments of the protein, they are moved into the endoplasmic reticulum via the TAP transporters. Once inside, these protein fragments are loaded onto the MHC molecule. So if I make a little tiny MHC molecule here, these protein fragments will be loaded here. Once the protein fragment are loaded into MHC1, that MHC1, so this all is happening here, right? So finally that MHC1 will be put in a vacuole. So if I make the MHC1 here, it will be put in this vacuole, and the vacuole would then transport to the cell membrane, and eventually on the surface of the cell membrane we would see the MHC class 1 molecule with the peptide fragment on it. So that is a normal process. Very quick recap. The important thing to note, so we have understood so far this much, that number one, all nucleated cells exhibit MHC or express MHC class 1 molecule on the surface. Number two, normally the MHC class 1 molecules, they are loaded with the protein fragments, which are usually the proteins of our body, cytosolic proteins. So there is no problem when these proteins are broken down, their fragments are exhibited on the cell surface. Does it do anything? No, it does not. Cytotoxic cells, or the cells which work with the MHC1, remember CD8 cell or cytotoxic T lymphocytes, they are the one which interact with the MHC class 1 molecules, and they would then, depending upon the situation here, they would react to the cell and do something. So I'm not going into that detail yet. I'm still focused on the MHC class 1. If the class 1 molecule is loaded with the peptide fragment of our body, then there is no harm to this cell. There is no danger to this cell. Good? So far so good? Okay. Now let's understand. In our initial lectures we had discussed this, that two situations cause reduced expression of MHC1 on the surface of the cell. What are these two situations? Remember that big picture and that house which was sick? So a cell, do not forget this. Why will MHC1 be reduced in a cell, on a cell surface? That is the question. When? Two reasons. One is viral infection, and the other one is malignancy. Viral infection and malignancies. So what happens is, when a virus jumps into a cell, so let's say the virus came in here, right? So this is a virus sitting here, dancing, having fun. Hey, I'm inside the cell. What is it going to do? This virus is going to steal the cell's machinery and start making viral proteins instead of the cellular normal proteins. Those viral proteins which are formed will also undergo the same process. Some of those proteins will break down. Some of these proteins will break down. Those broken down fragments will be transported into the endoplasmic reticulum via the TAP. Then they will be loaded into MHC1, and the MHC1 would go out on the surface. Now what will happen? If there on the surface we have protein fragment which is not that of our own body, which is foreign protein piece, then the cytotoxic T lymphocyte would attack. They will detect this thing and they would attack this cell and they would kill it. So what do you think virus does? Virus be very attentive here. This is something which you need to write down. Viral infection. Virus tries to reduce or downregulate the formation of MHC1. Very important thing. Virus downregulate the formation of MHC1. What is the benefit? If MHC1 are downregulated, then there are less MHC1 on the surface of the cell exhibiting the virus proteins, and there is less danger from the T lymphocytes. Very cool. So that is a way virus is trying to defend itself. The other thing, malignancy. Malignant cells also do a similar thing. They also cause the cell machinery to become different, and they also downregulate the MHC1 expression. Cool? So two reasons, only two reasons, there is no third or fourth or fifth reason so far. Only two reasons, viral infections and malignant cells. They downregulate the MHC1. So now we know that MHC1 will be interacted with the cytotoxic T lymphocytes, and they would kill the cell if necessary. But if the MHC1 is downregulated, this cell becomes protected. If it is protected, then who is going to help? That is the question. The question is, we have found a cell, we have found a sad cell, we have found a sad cell which has less expression of MHC1 on its surface. Which cell, which immune effector cell will take care of this? That is the question. So if I make a normal cell, this is a normal, healthy, happy cell, it has a lot of MHC1. It has a lot of MHC1. Compared to that, this malignant cell or infected, viral infected cell has less MHC1. Remember now, so let me give you a hint. Remember I said a pervert, a cell which is pervert, it is part of innate immune system. It massages, it touches, it handles every cell. It goes to the cells and goes and touches it. Do you know what that cell is? That pervert cell is natural killer cell. Natural killer cells, I'm going to laugh with this pervert cell. Remember, I look at the bigger picture lectures. Natural killer cell, when that cell touches the other cells which have gotten less MHC1 on them, what will happen? They would detect that the cell is there, and they will, what will they do? What does a natural killer cell does? So sorry for my little giggly fest there. Well, when the natural killer cell finds out, it touches the cell and figures out that there is less MHC1, it says, oh my God. So what it does is it sends perforins. Remember we talked about those. So again, I'm not here to revise the cell itself or natural killer cell's function, but generally they send perforins. Perforins cause holes in the cell membrane, and then they send granzymes, gran for grenades, granzymes, which go into the cell and they start apoptosis of the cell, or they encourage the cell to suicide, to kill itself. So that is how a malignant cell or a virally infected cell will be killed. So now, very important, neutrophil, they do not do this. Neutrophils are part of the innate immunity. They would pick up bacteria or a pathogen, virus, bacteria, toxins, those things, engulf them and take care of them. So what do? They do phagocytosis, they do pinocytosis, and they send nets outside. Remember, that is the latest studies that neutrophils cast nets in which they get pathogens trapped. So neutrophil is not going to take care of a cell which is sick. They don't do it. Macrophage, similar behavior. They do not take care of the cells which are sick. They do take care of the cells which are broken. They do take care of the debris, but they do not engulf the cells normally which are virally infected or malignant. Cytotoxic T lymphocytes, yes, they should have taken care of this situation, but the problem is cytotoxic T lymphocyte needs MHC1 to be present so that they can detect the foreign proteins and then take care of the cell. So for cytotoxic T lymphocyte, this is very important, don't forget this. For cytotoxic T lymphocyte, MHC1 expression should be normal and it should have the foreign proteins on it. So in this particular question, we are saying that there is a downregulation of MHC class 1. Cytotoxic T lymphocyte will not do anything now. And so natural killer cell, the perverts, the one who are going to massage the cell and then detect the less MHC and then kill that cell, that is the answer. So the answer is this. Very good. Thank you very much. We'll continue with our lectures.